Hello, biologists, and welcome to your latest installment of virtual lessons on DNA. Today's lesson from section 8.3 deals with DNA replication, how DNA copies itself. And where we last left off, James Watson and Francis Crick had just made a fabulous model of DNA. And one of the things that Crick had said right away was, looking at the model, it tends to give a hint as to how we think DNA copies itself. Because of base pairing, it really makes it pretty simple for DNA to make exact copies of itself. So let's talk about this copying process. We call this DNA replication, and it happens during the cell cycle. If you remember back to the cell division chapter, the S phase of the cell cycle is where the DNA copies itself. It does so that each new cell is going to get an exact copy of the original DNA, the original instructions in the cell. And so during the S phase, this one original copy makes itself into two new copies. The single DNA strand is a template, so it's almost like a mold so that the new strands are going to be exactly the same, and that makes sure that the information that each new cell gets is exactly identical to each other if the process happens right. So how does it work? So first of all, we have a double-stranded model that we have to expose these bases in between. So it unzips. It really is like a zipper unzipping, except a normal zipper would unzip at one end and unzip all the way through it. This is like a zipper that zips in the middle, and so it breaks apart in the middle, kind of in the model shown at the bottom here. And there's multiple points where it does the unzipping. So it starts at several places. It unzips as they zip towards themselves, and eventually you end up with two unzipped strands of DNA. And there's an enzyme which does this. It's called helicase. It comes in, breaks the hydrogen bonds between the two strands of DNA and unzips it, just like a zipper in the middle. The next thing that has to happen is we have all these exposed bases, A's, G's, C's, and T's that don't have a partner. They don't like that. Now, it just happens to be that floating in the cytoplasm of the cell are these free floating nucleotides, A's, G's, C's, and T's, and they're attracted to and bonded to their complement. So a T is going to find the nearest A and attract to it. The G is going to find the nearest C and attract and bond to it. And so the new nucleotides simply attract and attach themselves to the exposed bases of the unzipped DNA. Each strand kind of acts as the mold, the template for making the new molecules. When they're already on there, then an enzyme bonds the nucleotides together into a chain, uh, which is called DNA polymerase, and it makes it basically like a full DNA molecule. Each strand is complementary. Again, that means that they're not identical to each other, but they're the complements. T's are on one side are complemented by A's on the other side. C's on one side are complemented by G's on the other. And we end up with two full DNA molecules that are identical to each other that are coming off of these replication sites. And so when we get done, we end up with two molecules. They are identical. And there was a while where they didn't quite know how these molecules were. Are they conservative? Conservative means the original molecule acts as a mold, the new molecule is made, it breaks free from the mold, and the original is still in its original form. Or is it semi-conservative, where the molecule breaks apart, the new part is added to the old part, and each new strand consists of one part from the original and one part from the new? That's called semi-conservative because you're conserving half of the original molecule in it. Or is it dispersive, where there's different parts added at different places, and it would look something like this, where there's new and old pieces kind of making up each chain. And what they discovered was some testing and research. They found that DNA is semi-conservative. Half the original strand and half the new strand make up the two identical strands that are produced. And a short video kind of shows how this process happens. And so if we can, we'll take a look at this. And this is going to show uh, So there you go. That's the process of DNA replication. And again, it is semi-conservative. In the end, you're going to have two identical strands so that each new cell from cell division is going to have exact copies of the instructions. So hope you enjoyed today's lesson. Um, we'll have one more installment before this is over. Thank you for watching.